Hello everyone, and welcome to today's webinar, High Parameter Sorting and Analysis Using the Bigfoot Cell Sorter. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by ThermoFisher Scientific. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Alan Saluk, Senior Scientific Director, the Scripps Research Institute. Alan, you may now begin your presentation.
Hey guys, this is Al Saluk coming here from Scripps. All I can say is initially to start out, I'm not the biggest fan of webinars. I love seeing all your guys' faces and the direct back-to-back and the recognition of whether what I'm telling you is being processed or there's something that obviously needs further elaboration. It's really hard to do that when you're staring at your own computer screen in your office. So bear with me on that. I know at the end we'll have some questions. And before I actually start this talk, let me just pop a little background into how I'm structuring this talk and what I want to make it about. Initially, when I was asked by Thermo to give this talk, I was really worried that I was becoming kind of a walking spokesmodel for a piece of instrumentation. And I didn't want to do that. I didn't want to just be a glittering brochure, but rather I wanted to tell some kind of story. And it turns out, as I'm telling this story, it's a long story. And I'm not sure that all the parts of the story can be told within this initial talk. But first, I want to frame it in the context of what my history with flow cytometry sorting is about and how we've gotten to this spot in flow history where we're doing sorting based on spectral signatures. So I thought in telling that story, it would be best if I told it through the evolution of all the different cell sorters I used over the decades and how they've led to where we are now. Because each of these instruments at the time had unique stellar features that were then carried forward in the next set of instrumentation. And along that, it also tells a story about how I ended up using different instrumentation from different manufacturers, what their pros and cons were, and why we're using what we have now. So with that in mind, I have here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different little epochs of instrumentation that I want to talk about. And I'll go through kind of each one describing how it helped us at the time and what some of its cool traits were. So I originally came to Scripps in 1998 under the legendary Joe Trotter, who many of you know, and that was quite big shoes to fill, both intellectually and literally. He is a big guy. And I had a great time under him before he went to BD. And I thank him tremendously for helping open these opportunities for me. So when I came over to Scripps from Coulter in the late 90s, I was trained to use the Epix Elite, which is very similar to the BD Fax Vantage. And their configurations were 3 laser systems. And the lasers were a lot more different back then, to say the least. They were large water-cooled ion lasers that required significant infrastructure and they required tuning, but they were really powerful lasers with great beam qualities, and you could tune them to different colors. It gave you a lot of inherent flexibility in your systems. And the original Vantage was an 8-channel system. You had the classic 2 scatter detectors forward and side, but you had 6 PMTs for fluorescence detection that could be configured off of the 3 different lasers. And that configuration also was another degree of flexibility within the system. You could use different dichroic filters and different band pass filters to use different reagents that were excited by the different laser lines we're using. So you didn't have a real fixed configuration. You would typically adjust the system to optimize it best for the user who was coming to use it. And that was the norm for a while. We're all using the CellQuest software, but this was back in the days of what I want to call the LP record days. We're still using analog systems. And just like an LP record where you kind of hear the little hisses and scratches in there, there was kind of built-in noise to the system in the log amplifiers that just gave a certain visualization effect to data, especially during compensation. And we didn't really realize that until in 2000, we had the prototype BD Hyperfax. I actually couldn't find a picture of it because I think it's been buried in the annals of flow history. But it was basically a modified fax Vantage that had digital acquisition interface such that we're now taking the pulses and digitizing them. And I think at the time it was a 10 megahertz system. And we had LabVIEW as the software to use. If any of you use LabVIEW, it was super unstable. But off of those 3 lasers, we were now able to do 8 colors. And that was a first. And I remember sitting, I think it was a 2000 Cyto conference in San Diego where Mario asked, who does more than 6 colors? And I was like the only one to raise my hand. And that kind of freaked me out a little bit. But that was the beginning. We were now getting into the land of digital. And we started to notice some very unusual things, especially when you compensated, because the digital system remained true to the math of compensation. So the phenomenon of spreading was never really visualized before in the analog systems. And all of a sudden we're seeing that. And we're like, what is this? But one of the quirks that was really interesting in the Hyperfax was there was no baseline restoration. If you compensated something below 0, that data just disappeared. No one really thought there was negative space in flow, which we kind of all know now is there when you think about spreading and bi-exponential displays. So then came the first official digital system, the BD Fax Diva. Diva, I guess, stood for Digital Vantage. And it was a 3-laser Vantage. It was pretty cool because it had a switch that you can go between digital or analog, depending what you want to do. But along with it came some other cool evolutionary features. One was 4-way sorting. Instead of the classic 2-way sorting, you could divert up to 2 different populations to each side, giving them various degrees of charge to affect their trajectory. And then you had the digital compensation, which was first really shown in a full commercial setting, a product that everyone could use. And that raised a lot of questions in and of itself. I remember when there was another really prominent lab that did a lot of flow here at Scripps that also had a Vantage, but they didn't have it digital. And when I first started showing them some data, especially looking at some of the longer reds, the PE size 7s and the APC size 7s, where spreading after compensation was very evident, they were just like, what is this? You don't know what you're doing. You're just shilling for a company. This doesn't make any sense. They were just so ingrained in the classic visualization of analog compensation that they initially couldn't wrap their head. And I remember them telling me, oh, you're just a shill for BD. And I was like, whoa, no, I actually think this is right. The math, I understand it. Just because this is the first company to do it doesn't mean you should poo-poo it. And that kind of taught me a lesson there. It's like, wow, people are really ingrained in their habits, and it takes a lot of energy to change them. And so finally, once the education spread, I was giving lots of talks on digital versus analog to explain it to people and explain the new bi-exponential displays and stuff like that, people caught on. But that wasn't the only cool features with the Vantage. One of the great things was the drop delay was automated. And there's a couple different ways to do it. But back in the old days, we basically sort puddles of beads, or people would do the 10 drop method. There's all these different, very manual methods. That was just one of the big chores of setting up a sorter. But for the first time, BD had their sort check beads where you could deflect, look at the total amount of deflection versus what was in the center stream and say, hey, I've optimized my delay because I can see the deflection is working. It was a great visual way to look at it, and you'd use your eye. And with these systems, we got up to 8 colors. So that was pretty awesome. We were impressed with how well this was starting to work. And digital eventually did come on, but these were still classic open architecture sorters with a lot of hands-on and required a lot of input from the operator. Then in 2002, this was a big game changer, the fixed alignment sorter. There was a sorter you could just walk up, turn on the button, and it was basically almost ready to go. It used that same kind of timing for the sorting. You'd run QC beads. It was just a really, really cool system. And it had fixed alignment with this hybrid nozzle that the cell went by a cuvette before it was spit out at the tip. And it was a full digital system, and they were getting more and more advanced. Our first one was a 10 color with 3 lasers, and eventually these things matured to 5 laser systems. But we discovered something else in doing this too. And this was really strange. This was another great lesson for a core manager like myself to learn. When we moved some experiments from the Vantage with its classic jet and air novel to the Aria, I had a researcher come up and say, wow, this sort looks great, but my cells weren't proliferating the same way they had before. So they're alive, but something slightly different is impacting them. And I'm like, wow, that's weird. And so we actually did a really elegant experiment comparing jet nair sorters to the ARIA sorter at the higher speeds, and noticed that, oh my God, this is true. We did CFSC staining for proliferation, and it turns out that there was something going on. And later we recognized that, even now there's publications, we call it sorter-induced cell stress. And we did gene profiling to show, sure enough, it turns on a lot of genes if you sort something in conditions beyond what it was meant for. But my big lesson was, wow, you can't tell a big multinational company that their flagship product doesn't work like they're expecting it. And I caught a lot of negative vibes by presenting that to BD. And BD, in their own way, acknowledged it by making a change to the design, but they never really acknowledged that, yes, I had seen something. They tried to say it was my fault and blah, 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 but they, behind the scenes, slowly changed the design. And the machine didn't have that problem as much anymore, but there was still something there. And it took an extra level of kind of experimental design introspection to make sure weren't sorting your stuff at too high a pressure. Okay, so this was awesome. The other thing about this machine was it initially signaled the end of the sort operator, because when they initially had it, they were saying, oh, you can walk up, anyone can use it, one button. Turns out it wasn't. There were so many little quirks about the initial design that was problematic that it took them years to get it working sweetly. But it was definitely a game changer. It was a super sensitive sorter that was really easy to set up. That was cool. But because the researchers at Scripps couldn't recognize that there were some proliferation issues, we ended up getting another newer digital system. We took the Cytomation MoFlow platform, and after Beckman purchased them, and we were able to repurpose our big water-cooled lasers. It was really a less expensive way to move on to a newer platform, and we started using these systems, and they were really cool. I mean, they continued with the multi-laser with the spatial delay. They still had the digital compensation. They did the sort drop delay in a slightly different method. They didn't use beads, they used imaging. They basically took pictures of the whole stream from interrogation to last droplet and computed how long that delay was. And I didn't believe it at first, and it took a few months of just manual verification doing the old puddle methods. But sure enough, that approach does work. So there's a couple different ways to skin the cat of timing. And this machine also, open architecture, 12 color, configurable, you put your own filters in. It really was a great workhorse machine. Okay, but then technology was evolving. Along comes the first, I want to say, turnkey automated sorter, the Propel Avalon at the time, or now it became the Biorad SE3. And that was a 9-channel. It was super automatic. Like, you literally turned it on, loaded some beads on, and it QC'd itself, timed itself. It had little servo motors that aligned the nozzle. It was pretty cool. And my original thought was this would be really popular on the user base for self-service. But it turned out it was just not capable enough to become popular in my lab. There wasn't enough channels. There's only 2 lasers. They were collinear, which is something I'm not a fan of. And you couldn't sort a full plate. A lot of people are doing the basic one-color sorts or sorting on the plates. But this one had strips. And while it was cool, it didn't really fit into the workflow of our lab. And I ended up donating it to the San Diego Zoo, where I believe they're sorting rhino sperm on it. But then along came the Beckman Astrios. This was basically a repackaged, evolved version of the MoFlow, but with a lot of really cool features. First, it was 5 lasers, all fiber fed. So the alignment was much more stable. It had the same drop delay by image processing, but one of the other cool things was it kind of had an integrated QC system. You would tune it, it still had manual setup required, but it would tell you you've passed QC in all these channels, so you set it up correctly. And then you can go ahead and run the drop delay automation. And the other cool thing about this drop delay automation, there was actual feedback that it would adjust itself and try to maintain its break off and alarm you and shut itself off if it did. That was pretty cool. And the next cool feature of this one was the 6-way sorting. Before I talked about 4-way, now you can sort 3 different charge levels in 2 different directions, gave you 6-way sorting, which is a nice tool that some of our immunologists were using. But the thing I liked most about these 2 systems and the MoFlow before was the improved plate deposition. It was a very smooth piece of engineering with like really fine stepper motors that was very accurate. And that combined with sorting straight down, instead of sorting a deflected droplet. We instead would bend the entire waste stream to the right and just sort whatever we wanted straight down, made it super accurate. So we can start doing 384 well plates. This was really cool. So this is one of the features of this piece of instrumentation that was, really useful in our hands in the Corps. Okay, so we used these guys for a while. And then the next system we ended up purchasing was 2 of the Sony systems. And I remember when Sony first came into Flow, and I was at one of the Cyto conferences, I was like, oh my God, the rounding error in Sony's budget is probably bigger than, all the other Flow companies combined. And they made a pretty good entry on the scene. And they came out with the first order SH. They came out with some very slick products, but they weren't super high-end and complex. Okay, in this case, we ended up getting the Sony MA900. It has 4 lasers, 2 are collinear. That kind of limits some of the approaches we can do it. And it's the first one to have disposable chips. And originally, people were kind of peeved by that a little bit. But since we bought 2 of them, it turns out you could use 1 chip on the other machine. You could double the life of your chip usage. And that worked really good. In the end, you're only spending 10 or 15 bucks a setup to run this. And that worked pretty good. I was hoping to make these user accessible machines like other labs have, but the user base at Scripps is just, I don't know if they're just lazy or just so in love with our service that they would rather have us do the work and just drop their samples off. So this machine's really nice, but what made this machine super cool was the finish. And I think it came from the Sony consumer electronics philosophy that everything worked really nice. The automation, the finish, the fit, it was all really cool. The workflow was really cool. It was a well done piece of instrumentation. Now, it didn't have all the traits of the previous instruments, so it just wasn't perfect. So what I'm going to do next is kind of go through those little traits I talked about and tell you what I think I've watched over that first decade and a half. I mean, having spatially separated independent lasers is a great thing. Having configurable filters and detectors was a cool thing too. That allowed us, with researchers who want to use novel dyes and stuff, to kind of optimize the system for that. Digital signal processing, I mean, that's just the way it is. 20 years ago, we looked at that like, oh, this is some dark magic that I don't know what it is. And now it's totally the norm. So I know given enough time, the user base is capable of evolving to newer technologies. The automated QC and automated drop delay determination, that's all really cool. I mean, you basically want to be able to turn your machine on, throw some beads on, and let it go. Okay, the robust plate sorting, the use of 3D4 well plates, I mean, that's kind of key for a lot of our research. A lot of high throughput screening groups want to be able to prep a lot of their compound studies by seeding a bunch of 3D4 well plates with cells. And this system works pretty good. And also finally, like the slick finish to make the instrument look actually nice, to make all the interfaces and the ergonomics really nice. I mean, those were all these traits that led to where we are now. And where we are now is, and the whole point of this discussion is, at the time it was the Propel Bigfoot. The initial discussions were in Fort Collins in 2019. When I came in, I called this instrument the love child of the Yeti analyzer and the Avalon sorter. It had a lot of the high dimensional features of the Yeti, and it had a lot of the automated features of the Avalon, and it was all put into one system. So it was all the inherited traits I described previously, and it had a pretty cool thing. At first, I wasn't too sure about it. It had this sample carousel where you could load multiple samples in there. I'm like, oh, who really does that? You know, why don't you just, it's not that big a deal to put a tube on, but actually it turns out it's pretty cool. When you're running a bunch of controls and stuff, you can just load them in there and hit next, 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 and not have to stick your hand in there a bunch of times. That's pretty cool. It maintained a great plate deposition system. And for us, like I said, doing sort single cells into wells or seeding 3 to 4 row plates, that's like an important part of our workload. So it works great for that. And it had 7 lasers and 56 detectors. And right away, when I saw that last one, 56 detectors, I'm like, wow, that kind of jangled something in my head that I'll get to in a second. But more than that, there was 2 new features I hadn't seen before that I now realize are kind of should be key going forward. One was the integrated biosafety hood. I'd never seen that before. We were at the time a couple of years into what I call the hood fetish, where every single sorter had to be jammed to the hood. Sorters didn't like being in hoods. The hoods were ridiculously huge and expensive, and it just didn't make a whole ton of sense. Yeah, I can't argue that it nominally increased the safety of doing this, but I feel like good PPE practice and all those things before, it was never really that big an issue. And then to suddenly be decreed, oh, every sorter now needs to be in a hood if anything's even remotely BSL more than one. Okay, so in this case, the actual whole sample handling area is encased in its own hood. If you look here, I actually showed you, we had the TSS people come in and they actually certified it. So this is a functioning laminar flow hood, which is just a boon for safety. And the fact that it fits into a relatively small footprint instrument and doesn't affect access and serviceability, that's just awesome. I didn't think too much about it at first when I saw it, but now that it's actually part of our lab and our workflow, it's great, and I really like it. And another thing that I never really thought of, and I've seen a little bit of before, but didn't appreciate until I really got to use it, is servicing the instrument. Okay, let's face it, service is a nightmare. Every single time a machine breaks, they all inevitably do for different reasons, you have to tell the service engineer, they have to come. A lot of times they come, diagnose the problem, don't have all the parts, got to wait for the part to get shipped. You wait for the part to get shipped. Oh, where is it? Is it receiving? Oh, it's on its way. You just sit there twiddling your thumbs for hours. And these poor guys are just overloaded with work, and it's a really slow process. But now with this new remote interface, basically you can sit on, whether it's Zoom or Team or whatever one you're using, on the work screen of the sorter and talk to those guys as they remotely get in and interface with the machine and look at what's functioning here, what's not functioning there. They have this whole super complex user back panel that allows them to diagnose the machine remotely. And a lot of times they can fix it without ever even coming in, or if they do come in, they already know what's not working and the part's coming in with them. And so with that, I think that has to be a trait that is a part of all future instrumentation, because the amount of time we've lost waiting for service just as a function of reducing, I'm making air quotes, optimized manpower and service is a burden on the researchers. Okay, so now I've talked a lot about where I was when I first saw this machine. I'm like, that's pretty cool. But I'm going to digress a little bit, because a few years earlier, there was what I want to call a disruptor in flow cytometry. A few years earlier, we had spectral cytometry really mature. Okay, so spectral cytometry was kind of already there. Sony made a spectral cytometer that was kind of cool. It was more of a novelty. It wasn't super capable. There wasn't a lot of lasers. But when I saw the Aurora came out, I'm like, I think this is where flow is going. And in 2017, it says I purchased a 5-laser 64. I actually prepaid for the 5-laser and it was delivered a 3-laser until the whole 5-laser system was ready. But even with the 3-laser, it was evident that, wow, this is really where we're going in flow for high-dimensional stuff. For 15 plus colors, it just looked great. And the workflow, the software, everything about that machine just kind of works. Okay, and then finally, when they had the full 5-laser system, it was able to do 30 plus colors, and I realized, oh my God, this is the mass cytometry killer. There was a lot of pressure from some different groups to purchase a big mass cytometry system, but those are their own can of worms. You're going to end up buying lots of specialized reagents. You need a significant infrastructure to deal with the heat from the plasma torch. It is a big, complicated system, and it had its own inherent issues too. But now that I saw the potential in the SciTech, I remember there was one PI who came over. I'm like, look, I know you really want a CyTOF. Give me a little bit of time. Let's see where this does. And right now, there's some of the biggest users of spectral flow, and it's really amazing. And right now, it's proliferating across the field. But the reality is, our analytical capabilities rapidly outpaced our ability to sort. And that's a big deal. As I was getting people who are doing 25, 30 colors, they're like, oh, I really wish I could get this population. In some cases, they're able to work on their panels and drill it down to something in the 15 color space that we can do on one of our existing sorters. But they really wish they could do the whole big picture, where you could be able to take this super high dimensional view of a sample analytically, and then if you need to, sort it. Okay, so now that I have this in place, let's go back to when I was first looking at the discussion in Fort Collins. When I saw 56 detectors, I'm like, oh man, that's only 8 less than the Aurora analyzer. And sitting in that group of people at that Propel meeting, there's only one other person with any experience in Spectral sitting in that meeting, and they had a Sony one. And I'm just like jumping up and down in my chair. I'm like, why is this system not Spectral? Come on, come on, come on. This should be considered for Spectral. And it was just crickets. They were like kind of giving me this weird look. And I'm just like, okay, whatever. So I guess for whatever reasons, at the time, they couldn't tell me they wanted to do Spectral. So Tidar, who was in charge of Propel, comes over to San Diego. And over the most delicious falafel lunch we had at this great little place, he tells me, yes, they've been wanting to make that thing Spectral, but for whatever reason at the time, they didn't want to say it. And so he offered Scripps to become a beta location. And I'm like, this is awesome. The timing couldn't be more perfect. We have people who are just right in position to do this. And we're all ready to go. And then COVID happened. And that obviously had other scheduling implications. And because of that, we had to put a lot of things on hold. And it was painful. They delivered it just in like early spring, like maybe 2 months after all the shutdowns happened. It sat in crates forever. They weren't letting anyone inside of Scripps. The research inside of Scripps became painfully slow as it went to just sustenance level, like mouse colony maintenance. And then once things started to ramp up, it took a long time to actually get the instrument unboxed and running. And then the harder part was the researchers took quite a while to actually ramp up enough for this higher level stuff. So actually, where I went from having people just lined up ready to do these experiments, I had like a really slow start. But luckily, I had one guy who was in a great position. And it was our buddy Landon Edgar, who's now at the University of Toronto, and he had a great model. He actually came from a mass cytometry background, doing high dimensional stuff and making all the reagents. And he came over to Scripps having done no flow, and he was just brilliant, picked up on it right away. And his model was looking at carbohydrates on different phenotypes. So it was a great model because you could use classic immunophenotyping combined with also co-expression of these different carbohydrates, and it all correlated out really well in this high dimensional stuff. So here's a case of him looking at all these different cell subsets, and then all these different cell subsets will have different levels of expression of these different carbohydrates, and so you could cross-reference all this. And in this perfect world, we would be able to sort it all. Okay, so now Bigfoot's finally working. Let's get him to prep this. And now I'm going to touch on another part of Bigfoot I think is really cool. And it's the way its experimental design workflow is integrated in the software. I really was impressed by that initially. Let me just talk about that for a second. So the workflow, as you walk on the sorter, it's all done with automation. You basically log in the machine, hit startup. It'll tell you if your fluids are low or everything's low, whatever, you put the juice in it, hit one button, does alignment, QC, does the streams, optimizes the selection, does the break off, optimizes the timing and the delay, and it's all done in a one-click process. And it can be as black box as you want it. You can do it in one click and never see, or you can kind of get into the more service level menus and actually look at the camera images of all the different streams to make sure it looks good to you. There's potential that if you really want to go down the weeds, you can optimize the drive frequency combinations for better resonance and get it to be a super, super stable. But so far in our hands, we haven't been having to do that. The machine does a really, really good job of maintaining the stream. And that's basically the biggest deal of sorting. I mean, if your delay is off, everything goes to crap. And we have confidence that for the first time, that full level of automation is really, in one click, is really working great. And now there's a couple other instruments that have that too, and it's made flow a much easier, or flow sorting a much easier process. So next, let's take a look at the actual spectral workflow. So we had done the normal sorter stuff, but now we want to move into spectral space. And they have some cool features on this system. They had a panel builder. You go ahead and add your colors, and I'll talk about that, add your reagents. And it tells you how complex it'll be. You know, in a perfect one, no colors spill into any other colors, and everything's super perfect with literally no overlap at all. But we all know that isn't the case. So in this system, you fill in the information in the panel builder, run single controls for each one. And as you go through running the single color controls, it's like, that's a good control. we'll be able to make a computation based on that. We're going to give you a green check mark. You just go through the system doing that. And then the system unmixes it and plots it all out. And now your parameters are spectrally defined. They're no longer defined by a given filter. It's defined by the actual unmixed curve. And then we would go ahead and run the fully stained sample, set up our gates, and we did a 6-way sort. This is really a proof of principle sort. There was some of the material taken for downstream analysis, but this postdoc Landon then actually got a really nice position in Canada, and he has since moved, so we never got to do the full follow-up on this that we wanted. And that's actually one of the hardest parts of doing this kind of research, is being able to test a new platform in a way that's actually scientifically meaningful. I mean, you can do all kinds of cutesy experiments, but you actually need to be able to do stuff that benefits the researchers. Otherwise, it's impossible to get them to do it. Okay, so now I'm going to go through and talk about that sort workflow a little bit more. So here's our panel builder, and this is another really cool thing too. This is a hodgepodge of 23 markers that we did on this one, or 22 and live dead. And then they're from all different manufacturers. It's not like a given reagent is constrained. This machine can only use these reagents, which you've seen in other instrument manufacturers' designs. This is ubiquitous in that you can use whatever can potentially work, assuming that it gets through the next step. So here's what all those reagents look like in a big combined spectrum. It's a big hodgepodge. But more meaningful, oh, sorry, I did the wrong thing. More meaningful is the complexity computation. And here we can see, as we add the different colors in the panel builder, it's a little blurry, but I'll kind of read it to you here. It says, here we have a complexity of 1. A complexity of 1 means nothing overlaps with anything. So as we start adding other colors, the live dead, the BUVs, you see it start creeping up in tense little increments, and it works pretty good. And every so often you'll see a one where you get a big jump. So using BV605, we jump from 2.55 to 3.1. That's probably the biggest jump in the whole system. In some cases, if we go from, so we can hear from BV786 to using FITSI, that adds no complexity at all. So we go through and add these, and then using Alexa 532 adds no complexity. We go through and add these different colors, and in the end, we get a total complexity for the experiment. This is a good way, even before you run the sort, you can kind of assess whether the degree of overlap, the predictive degree of overlap in your experiment. And that's a great workflow. Okay, so once we have all this plugged in the system, the next step is we run all the controls. And as we run these controls, it's just a simple matter of we're running, in this case, beads and then cells for the live dead. It basically said, this is a good control, there's enough positive, boom, and we'll do that. And it goes through and it adds it into its little calculation and tells you a green check mark or a yellow warning or a red, no, that's not working. Okay, so I'm not gonna dwell too much on the actual experiment, because I showed this on the Cyto, there was a Cyto talk for Thermo, and I showed that. And this is all basic sorting 101. We have our gates, we have viability, we have waveform analysis to do the doublet discrimination. And then we just went through a whole series of gates where I was looking for the T cell subsets with CD3 and CD11B negative, CD3, CD48 positive, and then we could look for some of the other classic T cell markers of activated T cells on some of these other cells. So we can see, yes, this is the T cell population. We drew our gates. Going through, we do the same thing for B cells, NK cells, same thing. We get nice discrete populations. We had all our Ts, now we have all our Bs, and we have our NK positive cells. And then we did dendritic cells, we did CD11C positive, we did neutrophils with LY6G, and all of these guys cross-referenced with the predicted Siglec expression. I didn't want to pack this talk full of a bunch of plots about this actual project. All that can be shown later if you guys are interested, but I just want to show the proof of principle that this thing actually sorted. Now, this is what's tricky. Validating a sort is a whole nother project. I think running a sort and then rerunning it on the same machine and saying, hey, look, everything fell in the gates, that works great. I think that's kind of a road to painting yourself in the corner. A sort has to be assessed on a separate analyzer to make sure there's no inherent mix-up in your sorter configuration that's leading you astray. So what we did in this case was we did this validation on the Aurora spectral analyzer. And so our controls, which were a mix of the ultracomp beads from thermos and cells, were then run on the Aurora. The unsorted cells and sorted samples were analyzed based on the same gates we used on the Bigfoot. And initially in the talk I gave for cyto, two populations, dendritic cells and neutrophils, were not shown because there wasn't a lot of cells and there was viability issues. So for those specific subsets, but what happened was there wasn't a lot of dendritic cells, but I'll show you the ones we collected were pure, and the neutrophils just degranulated on setting. The other issue I have here is when we're doing these things, I can't shut down my lab to the other researchers to gain access to all the instrumentation. I basically was waiting in line behind other researchers who were using the Aurora to try to jump on so I could run our samples. So because of that, it didn't look as perfect as I wanted it to, but as I'm going to show you, it worked. Okay, so going through right here, we can see here's classic 4 and 8. Okay, CD4 wasn't one of our brightest reagents, but we still get a clean definition of where it is relative to the 8. And sure enough, when we look at the Aurora, we took that little sorted bit of cells, they came out as 4 and 8. I mean, it sorts fine when viewed on another platform. We can go through, that's 2 of the populations, let's go through another 2. I mean, when we look at the B cells, sure enough, they all sorted B cells. And when you look at a sort, okay, it's 96%. These are probably cells that died in the waiting. And this is probably the few represented impurities or maybe a little bit of passaging or something. But this sort worked. We're not seeing unusual distributions here to suggest that the delays weren't working right. And same thing here as we go over to the NK cells. Sure enough, on analysis, they sorted very well. One of the things is this. If you look to the right of the sorted population, we get a lot of cells that I believe were correctly sorted, and I think might be that spreading we see might be a function of some subtleties in the unmixing that we're looking at now. I'll come back to that. And here is the ones I didn't show in the original plot or in the original presentation. So here we took the dendritic cells. We didn't really have that many. And if we look at them versus a CD11B versus CD19, sure enough, there they are. We're getting no B cell contaminant. So the sort's working. These are cells that are just probably starting to die a little bit. And you can also look at the MHC2, again, CD11C, and sure enough, we still see the same relative distribution. I believe that sort worked. I just believe there's not enough cells in it to make a really pretty picture. But from a functional sorting point of view, that totally worked. And the least pretty one was the neutrophils. There just wasn't that many that lived, okay, because they were just sitting in an inappropriate buffer for too long. But you can see of the little bit that we had, they actually sorted cleanly. Once again, there wasn't a lot of B cell contaminants. And if we look at the LY6G, it was basically all positive for that. So I believe that those sorts work. So this was 6-way sorting done on a 23 color panel, and the analysis done on a separate platform. Okay, I think I'm one of the few people around that has the ability to do that. And because of that, there's a lot of pressure on me to explain how all this works. Okay, so I want to come back to where we're at so far. Okay, so some of the conclusions are really basic. Spectral sorting on the Bigfoot works. I mean, the workflow works really good, and the validation of the controls during the process of when you're running your controls on the system keeps you from getting yourself in a weird place. It's ensuring that your color choice and your controls allow for proper run mixing. So there's a built-in QC in the system. The target cells I showed you before were sorted correctly with high purity. I mean, the ability to look on a different analyzer and a different platform and say, yes, that works, that's really important to me. And the sorted populations, while there might have been some viability issues due to the way we handled the experiment, there's no real evidence of contamination with other cell types to suggest that the sort did not work. I 100% in utter confidence believe this machine sorts correctly. Now, here's the not pretty side of where we're at right now. I'm sorry, no, that comes next. Scratch that. Proof of principle experiments were performed independently. Panel design, reagent choices were determined independently. The instrument setups were just done by us. I basically sat with myself, one of my sort operators, and the postdoc, and we did it. We didn't have an army of support people and applications people, holding our hands to do it. It was done by us. And the automation of software has allowed for users to make these experiments ahead of time and know that they have potential to work just by virtue of plugging it in beforehand and assessing that. So right now, I'm sure this sorter works, and the workflow keeps you from painting yourself in the corner too badly. But future considerations are big. And this is where we're at right now. And I think this is where the next great drive in Flow will be. It'll be comparison of these different spectral platforms. Because for a while, it was all defined by Cytec with their Aurora. And that thing delivered amazingly. But now we have the Bigfoot, and now Sony has their new ID system, which is a super high dimensional spectral system. And the amount of effort and resources it takes to be able to compare these things is really limiting. There's not that many places that have more than one of each of those three systems, let alone three of them. So the idea of a great cross comparison where we can do one experiment and look at it in all three platforms and then debate the nuances in the unmixing platform or the unmixing algorithms, that's where we're going to be going forward. This is a really, really complex space. We've only looked through it through one lens until now. Now that we're looking through it the second lens, it's opening up a lot of questions. Okay, there are differences. We're seeing differences, both good and bad. Sometimes you see more spreading on one machine than the other, and sometimes the opposite is true for the other machine. Sometimes one machine looks better on one channel, but not the other. And is it a function of the hardware? I know Bigfoot uses photomultiplier tubes. Cytec uses photodiodes. And then I believe Sony uses its own special photomultiplier tube with its own dispersive array. So those are three different light detection methodologies. Are they having an impact on the differences? Reagent and panel cross-compatibility. That's the giant hurdle right now because everyone's designing their platforms off the Aurora. And I can already tell some reagents work better on the Aurora and some work better on the Bigfoot. And actually defining those and being able to make an iterative panel builder that works across both platforms would be a really good next step. And that's kind of what we're working on. And there could be differences in the unmixing algorithms. Okay, that's where we're digging right now. And I have some really cool data sets that I have just acquired recently in the past few weeks that range from 12 to 24 colors that have been done on both platforms. And now we all have to go through that data, and there are some differences, and we have to discuss why those differences are and whether it actually impacts the end results. And finally, one of the great ones, and this is happening even in the analytical side, what's the difference in the controls? Do cellular controls versus bead controls make a real difference in the way the data is unmixed? And does that issue propagate the more you use one or the other? That's what we're working on now. So I believe I want to call this part one of Spectral Flow. Part two will be looking at all these future considerations. And I have the data in my hands, but dealing with all this and spending the time to run these super elegant experiments is complicated. It requires researchers to do their experiments in triplicates to look at three different platforms. And also the addition of other sets of controls where it's FMOs or single biological stains, things like that, just to assess the differences across the different platforms, the unmixing. That's a lot of resources and time required to do that. And that's what we're working on now. And I have three good data sets now that I hope to show in the next talk, or hopefully I'll see all you guys face to face in a meeting in the future and can start showing this. And with that, I'm going to kind of wrap up here on thank my crew. Brian Seegers, the sort manager. He is just on top of making sure it runs. Brian Monteverde, who is my senior full operator, who has bonded with the Bigfoot, and his workflow through the software and his recognition of if things are right or not right has become kind of just next level divine. He's really good at it. And Alphonse, who helps keep the rest of the lab running while we're all doing this bleeding edge type work. And finally, Landon for the work he did with the Siglec research and what he helped to set up these initial panels. And going forward, I'm working with Luc Payton in the immunology department, and we have a lot of really cool stuff I hope to show you guys soon. And then finally, Bigfoot support. I can't thank TDAR enough for recognizing the opportunity to work with us. Angie Goldfein, who's basically been the mother hen of helping keeping all of us crazy scientists and researchers all coordinated and down a pathway towards productivity. Nick Rohrbacher, the mathematician who's doing all the work with the unmixing and helps us unravel when we have issues that are unclear. Mitch Durham, who's one of the lead engineers who helps us remotely whenever we're in trouble. One click text to Mitch, and he's up on the machine looking at it for us from his couch in Colorado, because we're usually calling him late. And then finally, they just got Kelly Gunston as one of their application people, and her knowledge of spectral panel building and spectral troubleshooting has been really useful to us.
Thank you, Alan, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question here is, have you noticed any differences in the spectral unmixing method between the Bigfoot and the Aurora, since you have both?
Okay, well, that's right to the point there. The answer is yes. And as I said before, originally we went big. Original first kind of replicative experiments were 23 and 24 color, and that's a lot. And as you start looking at little variances in that, you're not sure, is it panel design issues? Is it one machine or the other? And we're kind of trying to unravel this stuff, because we are seeing differences. In some cases, I'm seeing bigger spreading in some channels on the Auroras, and I'm not seeing it as bad on the Bigfoot. In other cases, I'm seeing stuff that's not as bright on the Bigfoot that looks great on the Aurora. So it's come to the conclusion that it's kind of an apples and oranges situation. I think a panel optimized for the Aurora is not going to be optimal for the Bigfoot, and vice versa. So right now, one of our current quests is to come up with maybe 15 colors that is going to work great on both. And that's what we're moving towards now. The 12 color panel we have that I'm looking at from the Teton Lab looks like it might meet that. And we're seeing some little subtle differences in the unmixing, and that's kind of what we're working on right now. So I don't know if I can answer that question. I can say, yes, there are differences. I can't answer them in full confident detail yet, because I know there are differences, but I need to be able to say why, and that's what I'm working on right now. And I think that is the germane question here, because SciTech has defined what spectral should look like, and we don't know yet whether maybe Bigfoot sees it different, or maybe there's some ground in between that's actually optimal, and that's what we're trying to figure out now.
Thank you, Alan. Our next question here is, since you used both beads and cells for the single color controls, did you find an advantage with one or the other?
Oh, yeah, another great question. And the answer is yes, with an asterisk next to it. I mean, no one can deny beads are the way easier control. Okay, they're bright and they'll do whatever antibody. The use of a biological control isn't always possible. Some epitopes just aren't expressed at high enough level to give you a good unmixing. Okay, they're going to be either too few or too bright at the higher levels. And you can't just swap, reagents, because the tandems aren't exactly the same. That really becomes obvious in spectral space. So right now, I think in a perfect world, and we have one group that's doing it, they're trying to do everything with cellular controls. But what we've done in some recent experiments, some of the data we have, we've done it with cellular and beads, and looked at it both ways, using the beads as the unmixing and the cellular, and to see whether it affects the unmixing of the controls. And that's what I'm working on right now. One of the issues with Bigfoot that I know is being fixed right now is that when you run your controls, you aren't able to visualize them afterwards. They're just basically processed in the unmixing algorithm, and that's that. You don't actually save them as a sample. So we've gone back and then run the controls as a sample, which takes a lot of work in a 22 color panel. And that's what we're looking at right now. Yes, I believe, I can't say yet, I believe there are subtle differences that have to be looked at, but I don't think one or the other makes or breaks an experiment. I think they can be used together, and I think as long as you're defining your autofluorescence with whatever cell type you're using, you're probably going to be okay. But once again, this is some of the bleeding edge stuff that we're trying to figure out now. I know Cytek's looking at that as well, and I think in some communications with Maria, she's like, in certain areas of the spectrum, beads and cells can behave a little differently.
Thank you, Alan. Looks like we have time for one more question. Any advice on transferring panels from your spectral analyzer to the Bigfoot?
Oh my God. These questions are like the trifecta of, oh my God, needs more work. I don't know. And that's what we're doing now. Like I said, we've hit on that 11 color. And I think what the next step to do would be in iterative panel building. You can go with what's going to work best. To a certain point, you're going to be able to get to what works best on both machines before you have to start bifurcate into adding this color will work better on this machine or to work better on that machine. And I can tell you right now, probably in 15 or 20 colors, you're going to start seeing a panel's going to work better for one or the other. So there's going to be a lot of work done. And unfortunately, I'm not capable of doing it all. And I know other people are kind of ramping up to where we are right now. There's going to be panel design optimization for the Bigfoot. I think right now everything's optimized for the Cytek, and we've been trying to walk in those same footprints, and it's just not quite the same. And so I think the work on us now for people who want to do the sorting on the Bigfoot is figuring out where the commonality splits and what reagents are better biased towards the sorter platform.
Thank you, Alan. Do you have any final comments for our audience?
Yeah, this is part one. This is, unfortunately, I was told I would give this talk. I said I would give this talk, and I'm sitting on a pile of data that would answer all these questions, and I'm dying to dig deep into it. And I think really these data sets will help answer those last three questions better than anything I'm showing you now. And I do believe Spectral is totally the way to go future forward in high dimensional flow. And I think the Bigfoot's an amazing platform. And I love the competition in this space between Cytek, Thermo, and Sony to all have these high dimensional spectral systems. And I'm impressed by what I've seen so far. And I think in the next couple of years, we're seeing the same growth we saw when we switched from analog to digital. Now we're switching from classical to spectral, and that's where the future is.
Thank you again, Alan Saluk, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, ThermoFisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.